are delighted to have our speaker, Kathryn Paige Harden, today. Kathryn got her Ph.D. at the University of Virginia in behavioral genetics. Actually, it's a hotbed of behavioral genetics there. And then went to UT Austin, where she is a professor of psychology and psychiatry, and has become a leader in the whole field of behavioral genetics. Has written a best-selling book, The Genetic Lottery, Why DNA Matters, which not only talks about a lot of the research in the field, but what's the implications of all that research for education and how we raise our children, and what are the society's obligations to people who didn't win one lottery versus or another, and has provoked a lot of discussion. I know Rebecca and I had some good discussions about Kathryn's book, and we're really looking forward to hearing from her today. Kathryn.
Thank you so much for inviting me to your lovely department. I do stand by the assessment that at least the faculty with whom I've met are above average, not just in intelligence, but also in extroversion and warmth, and I have thoroughly enjoyed my day. I wrote an abstract, which your lovely and very competent administrator asked for much in advance. Almost entirely talking about the abstract I submitted was talking about the scientific work that I want to share with you today. But since I sent that to her, I thought that I would do something maybe a little bit different and try to talk both about the work that I've been doing with my colleagues on the genetics of what we call externalizing spectrum disorders, but also try to put that work in a much, much broader historical and philosophical context and talk a little bit about some of the themes from my forthcoming book project, which will hopefully be out next year. Anything empirical that I'm going to be talking about today is very much not just mine. It's work I've done in collaboration with the Externalizing Consortium, which I co-lead with Danielle Dick from Rutgers, Abraham Palmer from UCSD, and Travis Mallard, who's here in Boston. And really the lion's share of the credit for getting these analyses done really lie with Travis, and then my former trainee, Camille Williams, who has unfortunately left UT Austin because for some reason she wants to live in Paris rather than Texas. I'm very confused about that. Okay, so to get us started, I want to think about an unusual place at an unusual time. I want you to think about an imperial power at the height of its military prowess that has recently embraced Christianity at the highest levels of government, and that is just a few decades away from collapse. And I am talking, of course, about the Roman Empire at the end of the 4th century, where Augustine of Hippo, who was raised in North Africa, was proposing an idea, a theology, a doctrine that was both a radical departure to what the early Christian church understood about human nature and human freedom, and was also turned out to be, I think, one of the most enduringly influential doctrines in the Western world, and that is the doctrine of original sin. So the doctrine of original sin, even if your experience with Christianity is only glancing, you've probably heard this phrase before, original sin. Sometimes translations render this as the doctrine of hereditary sin. And the idea behind original sin, well, I don't know if this was the conscious motivation, but it was a neat trick because it accomplished two things. And the first thing it accomplished is by describing what Augustine saw as the limits of human perfectibility, justified the necessity of this merger of ecclesiastical church power and state power that you saw at the end of the Roman Empire after Constantine's conversion to Christianity. And the second thing it did is it helped the Christian church make sense of why do some people come into the world already suffering, already different, so unfortunate in their circumstances or in their health? How could a just and good God have created children that suffer. And Augustine's neat trick was, well, they must not be innocent. If they're suffering, they must have inherited something that makes them punishable or damnable. Infants arrive polluted by sin, he wrote. Since they have committed no actual sin, remission must be for the guilt attaching to the faults in their nature. Now, I already mentioned that this was soon to be the orthodoxy of the Christian church, but it was not without controversy even at its time. One of Augustine's foremost intellectual opponents at the time hailed from the other end of the Roman Empire, the British monk Pelagius. And he was eventually condemned as a heretic. And the Greek word for heresy comes, or the original word for heretic comes from the Greek word for choice. And Pelagius insisted that for something to be sin, for something to be morally condemnable, to be blameworthy, it needed to be an act of freedom, which means it couldn't be something that you inherited. His follower Julian summarized it this way: whatever is natural is shown not to be voluntary. These two are, by definition, are opposites, like necessity and will. The Christian theologian and historian of the church, Paul Tillich, said that America, despite its Puritan roots, remains very much in favor of this Pelagian idea. The contradiction of a moral demand must be an event of freedom and not a natural event. So y'all are scientists, devoted to rational inquiry, and I lured you here with promises of GWAS. So why am I talking to you about early Christian history? And for 3 reasons. The first is I just want to note that the questions that they were preoccupied with, the question of what do we inherit, genetically, culturally, economically, they would have never phrased it this way, obviously, but evolutionarily, what is the relationship between that inheritance and how we behave, are still questions that many of us are still preoccupied with today. And our answers are necessarily going to be viewed and perceived within an intellectual framework that vastly precedes us. The second is that they were not just interested in human inheritance for purely academic reasons. They thought it had bearing on the question of what are we to blame for? And I know I'm not the only one who, by studying human behavior, has found themselves at times, their work being used in debates about blame, about responsibility. And again, that's because we're interrogating these questions within an older intellectual framework. And the third thing is I've begun to wonder, how do these ancient intuitions, competing ones, about the relationship between inheritance and behavior, how do they not just shape how our science is perceived by other people, but how do they also shape the science that we conduct and the way that we frame our research programs? Obviously, this is a question, what do we inherit and how does it shape how we act? That is something that I've had a lot of time to think about because my field is in behavioral and psychiatric genetics. And I want to pause here and think about, why would we want to understand genetic risks or genetic liabilities for human behavior, complex human behavior, or what we have defined as psychiatric diseases? So I think just like in medical genetics, we're often interested in understanding genes to improve our risk stratification. Who needs early intervention? I was describing earlier today that I went to the doctor and I had like slightly high triglycerides, and they were like, maybe this is bad, maybe this is okay. Why don't we get more information about you to see if we need to do something about it? That's a very common dilemma that we face in the clinic with child and adolescent patients. Is this, as one of my supervisors in graduate school called, normal response to effed up parents, or is this the beginning of a lifelong psychiatric disorder? That kind of risk stratification, who needs to be prioritized for early intervention, is a persistent clinical problem. We might want to improve power to identify causal effects of the environment by just kind of taking into account that genetic variation. I think oftentimes we're interested in identifying heterogeneity in response to interventions. Not everyone responds to lithium. Not everyone responds to Ritalin. Not everyone responds to family therapy or to parent training. If you pass a new law that requires people to stay in school longer, then that improves health on average, but not for everyone. It seems to work better for people who are at higher risk for obesity. There are individual differences, and there's also individual differences in who's being helped by the things that we're trying, and we might want to try to see some of that heterogeneity. This probably goes without saying for this audience, but we're interested in understanding mechanisms. We're interested in understanding why people come to develop certain outcomes, and genes can be a tool, genetics can be a tool for seeing that. And then also, I think it can be a tool for identifying novel intervention targets. You'll notice that what's not on this list is adjudicating between competing intuitions about what should be appropriately considered morally blameworthy. And yet, that is, in fact, one of the most common uses of genetics in the public square. And that's particularly salient for us because what we study is what has been termed externalizing spectrum disorders. So externalizing is a term that comes from the 1960s from the psychiatrist Achenbach, who was doing psychiatric case file reviews, and essentially then, as now, being dissatisfied with the diagnostic systems by which psychiatrists were labeling people in the 60s. And noticed that there was a constellation of symptoms that seemed to be what he called internalizing. So children who were sad also became anxious, and they occasionally became compulsive. And that there was also a constellation of symptoms that he called externalizing, which were things that got the child in trouble with the external world. So a child would come into hospital because they were pathologically setting fires, and then they would end up in hospital a few years later because they were addicted to drugs, and then they would end up involved in the criminal justice system later because they were stealing or involved in interpersonal violence. Decades of research since Achenbach was working have used twin studies, have used adoption studies, family studies, and pedigree studies. And in modern DSM nomenclature, what we see as the externalizing disorders, or what we label as the externalizing disorders, are ADHD, conduct disorder, and oppositional defiant disorder in childhood and adolescence, and then substance use disorder, and the cluster B personality disorders like antisocial personality disorder or borderline in adulthood. These are the diagnostic boxes that the DSM uses, but what we see is that they're highly comorbid across the lifespan. So a child with conduct disorder is 10 times more likely to get a diagnosis of ADHD than a child without one. 50% of children with conduct disorders will ultimately meet criteria for a substance use disorder, and you can't be diagnosed with antisocial personality disorder unless you had conduct disorder as a child. What we also see in pedigree studies is that these tend to run in families not just because of shared environmental risk. So if you have a parent, a biological parent who adopted you away at birth, who has a substance use disorder, you're more likely to get a substance use disorder, even if you weren't raised with them by them. But you're also more likely to be diagnosed with a conduct disorder or antisocial personality disorder. So what we see is, in some ways, like every other psychiatric disorder, a constellation of diagnostic labels that twin studies suggest are moderately to highly heritable and that are comorbid of the lifespan. But what's different for externalizing disorders relative to, say, anxiety or depression is that the hallmark symptoms of these disorders are also socially disciplined or punished behaviors. So children with ADHD are more likely to be suspended from school. Conduct disorder and antisocial personality disorder and substance use disorders are often defined by doing things that are illegal according to the state. So as soon as we begin to think, well, what are the genetics of externalizing disorder, we perhaps shouldn't be surprised that we're dragged into debates about blameworthiness. Oftentimes in psychiatry, what we see is what I would consider broadly Pelagian arguments, which are things that demoralize a phenotype via reference to the involvement of biological processes. So what I have here is an article from ADD magazine. Don't mistake your child's ADHD symptoms for bad behavior. Your child is not willful, disobedient, scattered, demanding, obnoxious, aggressive, or lazy. He or she has ADHD, a neurological condition. Within the DSM, the ADHD has been re-put into the neurodevelopmental disorders category. Okay, so again, I'm considering this broadly Pelagian because it is saying we shouldn't consider this morally bad behavior by virtue of the involvement of these biological brain-based processes. Perhaps the most famous example of this was Leschner's article in the late 90s, Addiction is a Brain Disease and It Matters. The more common view is that drug addicts are weak or bad people and willing to lead moral lives. To the contrary, addiction is actually a chronic relapsing illness. The gulf and implications between the bad person view and the chronic illness sufferer view is tremendous. And the data that he was bringing to bear in this paper was that substance use disorders are highly heritable, they run in families, and that they involve demonstrable neuroscientific processes involved in addiction. There is a limit, though, sometimes to Pelagian arguments, and that tends to be around antisocial violence or aggression. Landrigan was a man who committed homicide in the 1990s. He was one of the first to ask for his defense to enter in family history as mitigating evidence in his trial. He found out after he committed murder that his biological father, who had given him up for adoption, was also on death row in another state, also for committing homicide. And he asked his defense to make an appeal, stating that this family history was evidence of a liability to aggression that had been inherited and should have been considered a mitigating factor in his sentencing. It went all the way up to the Supreme Court, and every level the court disagreed. And at every level, they decided that even if his defense had introduced this family history, and even if the jury had been convinced that it was relevant to his likelihood of behaving in this way, that it could not have ever been viewed as mitigating. In the words of the Ninth Circuit, it's highly doubtful that any sentencing court, would have been moved by information that the defendant was a remorseless violent killer because he was genetically programmed to be violent, as shown by the fact that he comes from a family of violent people who are killers also. So the reasoning here is that even if this was relevant, it could have only made him more punishable in the eyes of a jury. It wouldn't be mitigating. There's some evidence to suggest that the court's conception of people's intuitions was correct. So this is data from Susan Gelman's lab relatively recently. And they used a kind of, I think, a clever setup to look at people's belief about the inheritability of certain types of behavior. They asked them to imagine that they had conceived a child with a sperm donor, and that after conceiving the child had found out various pieces of information about the sperm donor, and then asked them to judge the likelihood that the child that had been conceived would also show a behavior like the sperm donor had shown. So you can see that people think that if their sperm donor is tall, that the conceived child is also likely to be tall. Fewer people, but not zero, thought that if the sperm donor spoke French, the child would also perhaps speak French. And then you can see that the other phenotypes about the sperm donor were sort of intermediate between those things. IQ was estimated to be about as inheritable in this view as height, but committing an act of antisocial violence, in this case the sperm donor described as committing murder, was seen as maybe a little bit more inheritable than sexual orientation and a little less than being temperamentally shy. What's interesting is that they then asked participants to suggest what punishment they would recommend for these crimes that the sperm donor had committed. And people who thought that violent crime was more inheritable actually suggested longer prison sentences compared to shorter ones. The author speculated that this was perhaps due to some suspicion of sort of incorrigibility, and that in this case punishment was recommended for sort of incapacitation purposes. But this is working, in an opposite way, as we see for how often genetic beliefs about the genetics of, say, substance use or ADHD are interpreted and viewed. So when I first started talking about this, a common thing that I heard from people is, well, maybe that's because we found some genes, at least, that are associated with ADHD and with substance use disorders. But like, what genes are actually involved in non-substance related antisocial behavior? And if you do look at the record of previous genetic discovery, you will see that, especially compared to other psychiatric disorders, the record of finding specific genes associated with non-substance related antisocial violence or early conduct problems has been very, very, very bad. The last GWAS specifically devoted to conduct disorder was actually in 2011, which is like 900 years ago in GWAS years, right? And it included 872 cases. Since then, there's been one of comorbid CD and ADHD, again, in 4,000 cases. This is Domontis' work. And then an adult's ASB, which is not just antisocial personality disorder, but any involvement in antisocial behavior, 86,000 cases. I think this is really striking, right? This is considerably lower sample sizes and considerably fewer studies compared to almost any other psychiatric disorder that you could think of. And so we wondered, in part, in our externalizing group, is part of the reason we haven't found replicable genetic associations with antisocial behavior or conduct disorder is just we haven't looked hard enough. We haven't compiled enough data on enough people. But of course, compiling data on these phenotypes is difficult. Oftentimes, physicians don't enter conduct disorder into a child's medical record because there isn't a FDA-approved treatment for it. And many people who are involved in antisocial behavior are not involved in the medical system and signing up for studies. So there's a question here of people don't enter child conduct disorder into the medical record because it's hard to get reimbursed for it, because insurance companies don't think it's biologically based, so we don't invest any research in it, so there's no genetic associations with it, right? There's a kind of circularity here. Our strategy to get around this problem was to go back to what do we know? And what we know is that much of the genetic liability to any one externalizing spectrum disorder is thought to be shared with all of the other ones, and not just with the disorders, but with other forms of disinhibited or antagonistic behavior. So we thought, can we pool data on things that are genetically related to this core of disinhibition and antagonism, but by virtue of being more normative and less stigmatized, have been collected on many, many, many more people. Our first attempt to do this was published in 2021. And what we're using here is something called genomic SIM, which is basically a method to jointly analyze GWAS data on multiple phenotypes simultaneously, modeling their genetic relationship, and attempting to estimate SNP associations not with any individual one phenotype, but with a latent factor that represents a cross-cutting liability across them. So at that time, we identified 7 externalizing related phenotypes based on theory and previous twin studies suggesting a genetic correlation with externalizing spectrum disorders. So there's 2 sexual phenotypes, age at first sex and number of sexual partners, smoking, alcohol problems, ADHD, cannabis use, and risk tolerance, which is a completely garbage one-item personality item from the UK Biobank that asks, are you the sort of person who likes to take risks, yes or no? That they saw fit to ask in 600,000 people. So we decided the sample size outweighed the poor psychometric properties. And what we found is that using GWASs of each of these things, sample sizes for each phenotype ranged between 50,000 and 1 million people. They were all moderately genetically correlated. We could fit a kind of genetic factor representing that covariation, and we could estimate SNPs with that general factor. So we identified about 570 genomic loci that were significantly associated with that underlying factor. I'm not going to go too much into these details. We then took those beta weights, right? So a GWAS is estimating a linear regression of the phenotype on a minor allele count, 0, 1, 2. So for each of those SNPs, you're estimating a beta weight. We then took those beta weights into 2 independent samples, One is a population representative sample of American high school students beginning in the 1990s who have been followed until the present. And the other is COGA, which is a study of the familialism of alcohol use and related substance use disorders. So at least one member of the family has been diagnosed with an alcohol use disorder. And what we found is that a polygenic score estimated from the RGWAS results could predict more serious forms of disinhibited and antagonistic behavior in independent samples. So it predicted whether or not a person had ever been arrested, whether they had 3 or more criteria of conduct disorder or antisocial personality disorder, whether they're ever convicted of a felony, whether they ever developed a moderate alcohol use disorder, ever used illicit drugs, or ever used opiate drugs, and actually many other phenotypes, but these are the ones that I wanted to show you today. Now, obviously, these are not, this should go without saying, but I'm going to say it anyways. This is not genetic determinism, right? So there's no 100% minority reporting, know that you're going to commit a crime. These are, like every other risk factor that you see in population studies, which are small but meaningful in the long run associations with these outcomes.
Demographic factors?
Thank you. I will say that all of these people would be identified as white in the United States, and all were European-like in their genomic ancestry. Yeah. So we're not speaking in any way to racial differences, but also that's a limitation of our study because we're also only making inferences about like a very narrow slice of global genetic diversity. Thank you for that question. My postdoc, Peter Tanksley, and I also wondered about to the extent to which this was tapping uncorrected population stratification. To get at that, we're looking at data from the Millennium Cohort Study, which involves parent-child trios. So our within-family estimate here is, does the polygenic score predict child conduct problems controlling for the parental genotype? The between is the population association not controlling for the parental genotype. We then compared the effect size. So the answer is yes, it does. And then we compared the effect size to the effect size from other recent meta-analyses of epidemiological risk factors for conduct disorders in the US population. And what I just want to note is that it hangs, even though within family is commensurate with the magnitude of correlation that we see between conduct problems and, say, maternal smoking during pregnancy, peer victimization, lead exposure, or parental socioeconomic status. However, this was limited to people with just European-like genetic ancestry. Hold on. Our follow-up analyses suggested that our PGI was doing a decent job predicting disinhibited behavior, but particularly for measures of sort of callous and emotionality or violence and aggression, it wasn't picking up that part of the spectrum of externalizing as well. Obviously, better power discovery is more predictive polygenic indices. And we were talking about this today in several of our conversations. I think recent research is suggesting that even small genetic associations can give us meaningful insight into mechanism. The example I was using in a conversation earlier today is what we see for Ozempic and weight loss. So if you do a GWAS of BMI, you do get genetic variation in GLP receptors. It's not a huge signal. But if you push on that mechanism using something that's a way bigger intervention than you're seeing in the scale of naturally occurring sort of endogenous genetic variation, you can get a really meaningful effect. There is data to suggest that drugs that are making it through clinical trials are more likely to be successful if they have some GWAS support. And GWAS support is defined pretty minimally, right? And without regards to the effect size. So I do think there was a time where people debated if we're just throwing more power at something, are we just getting more scree at the bottom of the hill, right? And all the big important stuff has already been found. I actually don't think that's the case. I do think there's still juice left to squeeze in these analyses. Yes, Manolis had a question.
If you go back a couple slides, I was basically noticing the child maltreatment. I mean, this is one of those cases where unless you're looking at adoption studies, it's very hard to distinguish the environmental and the genetic effects.
So that's another thing. So all of these things, child maltreatment, maternal smoking, lead exposure, parental SES, these are correlations. that are estimated without causal identification, whereas the polygenic index within family is getting at what I would characterize as causal. So that's a good point.
Is that it's not even a fair comparison because, for example, child maltreatment, maternal smoking, etc., compounds also the genetics. It's not just environment.
Yes.
If you looked at the effect in an adopted child,
Yes.
my guess is that it would be much less.
I think it would. I think on the flip side, one of our reviewers suggested, well, that's each environmental risk. You've done a polygenic risk. What about a poly-environmental risk? And I think it's an unfair comparison to the genetics because it is these epidemiological correlations, but also an unfair comparison to the environment because we're comparing a conglomerate compared to each individual environmental risk. I more just want to say that, like, from the perspective of someone who's thinking about a study and they're like, what do I need to measure in this population to capture what are the best known sort of epidemiological predictors of this? I think that if you're including child maltreatment measures and child SES measures, just by effect size, I think the polygenic index should be at least on the menu for consideration there. Okay, so in our updated analyses, which are not published yet and still very much in progress, we had larger GWASs for several phenotypes and also an updated GWAS of cannabis use disorder rather than cannabis use. So this, I'll get to you in 1 second, a pooled effective sample size here of around 3.5 million people across these phenotypes. Yeah?
Just a clarification,
Yeah.
GWAS?
What? Oh, gosh, I'm so bad. Genome-Wide Association Study. Yeah.
Which is kind of using all the genes in the world.
Yes, I should have done this at the beginning. Yeah, so what we're doing here is other people have largely done studies where they have measured very many SNPs, single nucleotide polymorphisms, single DNA letter differences between people scattered throughout the genome, and done regressions where they're estimating what is the linear association between number of minor alleles at each of those spaces and a phenotype of interest. What we're doing is we're taking those GWAS results, applying LD score regression to estimate the genetic correlations between them, and then fitting a model in genomic SEM to see if we can identify a genetic factor that represents this liability that's across all of them. Thank you.
And could you clarify common versus rare?
Oh, yeah, and so it's all common variants that we're looking at here. I think our cutoff was 5%. So we're not looking at any rare variants of large effect. These are all common variants that we would expect for many reasons to have tiny, tiny effect sizes that are nonetheless maybe less tiny in aggregate. Great questions, thank you. Okay, so we did that same thing again. Now we're looking at SNP associations with this kind of updated, larger, more powerful externalizing spectrum disorder factor. Again, our first question was, okay, well, we've analyzed data from, like, do you smoke a lot of pot and have a lot of sex? Like, are we getting at things that are relevant to more harmful forms of disinhibition? So we had held out several phenotypes where if we had included them, it wouldn't have added new people to our analyses. And then estimated the genetic correlation between our externalizing factor and those holdout phenotypes. And the 4 strongest correlates genetically of our factor are antisocial behavior, opioid use disorder, substance use, and interestingly, because many people think of this as a more internalizing phenotype, suicide attempt. But again, I think is a manifestation of harmful disinhibition. So I think, again, we are converging, we're using genetic data on more normative forms of risk-taking, but a diverse set enough that we are converging on genes that I think are relevant for more clinically severe, clinically relevant, harmful forms of disinhibition.
I want to make sure I'm getting what you
Yeah.
did here. So we derived the polygenic risk factor using these other dimensions, holding out those 4 at the bottom,
Yes.
and it predicted the 4 at the bottom.
Yes, and then we're estimating the genetic correlation between this purple factor, or all of these are held out. I'm just highlighting. These are all the held out ones. I'm just highlighting the 4 strongest. Yeah.
It's cool that some of the held out ones don't.
Yes. Right. So for instance, we're not picking up on something that is just redundant with the PGC psychiatric cross disorder factor. We're not picking, this is not anorexia, right? So, which you wouldn't expect it to be. It's not, it is converging in the directions that you would expect with other kind of smoking phenotypes, but it's certainly not redundant with them, right? So we're seeing both convergent and discriminant validity, I think, in these genetic correlations. We also did this in 3 GWASs that focused on African-like individuals. There's much less data here, and we only had GWASs on 3 relevant phenotypes: alcohol problems, cannabis use disorder, and smoking. What we did is we first estimated in Europeans a reduced factor that just had these 3 and said, okay, are we converging on the same thing? We are. And then we estimated the cross-ancestry genetic correlation between the factors. That doesn't always work out to be close to 1, but in this case, it was statistically indistinguishable from unity. So the genes that we seem to be picking up on with this more limited African-like factor and the genes that we're picking up on with a much more powerful European-like factor with more indicators seem to be essentially converging on the same genetic signal. We also see that if we look at how the African-like factor and the European-like factor are correlated with other things, and they're not correlated in the exact same way, but there's a lot of concordance. We can come back to that if you have more questions. So we conducted a GWAS separately within each ancestry group. The pooled total sample size was around 4 million people. And then we conducted a multi-ancestry meta-analysis. And what we end up with is an extraordinarily powerful GWAS, I think especially relative to any psychiatric or behavioral trait here. So we're identifying around 1,200 genome-wide significant loci. Travis is in the middle of doing fine mapping for these, and we welcome input about that if you have suggestions. Again, we're always concerned with, we're picking up on a genetic signal, we have a really powerful GWAS, do we have independent evidence that we're actually getting at something that's relevant to the phenotypes that we think that we're getting at here? So we're doing polygenic index analyses and 5 samples. I'm going to focus on 2, those same 2 that we used in previous AdHealth and COGA, which also have sibling data. So this is in COGA. What I have in the light bar here is the population association between our updated polygenic score and the phenotype of interest. What I have in the dark red bar here is the direct genetic effect or that association estimated within families, so controlling for the sibling genotype or the parental genotype. So there's a couple of things I want you to notice here. The first is, yes, we are picking up on significant variation in tobacco dependence, alcohol use, antisocial personality, ADHD, suicide attempt, suicidal ideation, cannabis use disorder, and opioid use disorder. Second, with the exception of smoking, the attenuation when we go from population level associations to within family associations is actually pretty small. This looks a lot better than what you see for something like education. So it doesn't seem like what we're picking up on here is just uncorrected population stratification. And 3, these effect sizes are small in a plausible way, right? So we're accounting for between 2 and 5% of the variance, definitely meaningful at a population level, definitely commensurate with what we get from other social science variables, definitely not genetic determinism. We also see this for more minor health risk behaviors, and also for some things related to criminal justice involvement. So ever arrested, and then the AdHealth measure of violence and aggression are, have you ever gotten into a physical fight? Have you ever pulled a knife or gun on someone? Have you ever used a weapon in a fight? So it is, not just like a minor risk-taking that's happening with the violence and aggression. AdHealth has also a number of social outcomes. So we looked at college completion, educational attainment, GPA. Have you ever been suspended from school? Do you currently have health insurance? Have you ever been arrested? Your number of pregnancies? Have you ever been incarcerated? Your household income. What I want you to notice here is that there is some prediction, but there's also a lot more attenuation between the population and the within family models. So when we're talking about genetics predicting psychiatric symptoms or genetics predicting health risk behavior, we're seeing that we're picking up on, I think, good evidence of true genetic signal. But when we're thinking about genetics predicting these social and medical consequences, all of a sudden the picture looks a little bit more complicated here, and it does seem like there's less reliable genetic prediction when you're comparing people who are starting off in life in the same family. We then took our PGI into electronic health records. This is BioView, and we conducted a FEWAS. There's a lot to talk about here, but again, I just want to note that the top 25 associations of our genetics with any electronic health records codes are with tobacco use disorder, viral hepatitis, HIV infection, antisocial personality disorder, substance addiction, suicide, obstructive chronic bronchitis, post-traumatic stress disorder, alcohol-related disorders, chronic bronchitis. So what you're seeing is a lot of suffering, you're seeing a lot of despair, and you're seeing some medical consequences of behavioral risk-taking, right? So in terms of the medical consequences of this genes, I do think that we're seeing this borne out in people's health histories.
What do you think of the effect size? It's about like 1.5, 1.4?
Yeah, so it's between I did this last night, 1.3 to 1.5.
Is this much less than you would have expected?
No, I don't think so. I mean, again, we're talking about, a population health registry and an electronic health record. I mean, there's noisiness on both sides, both the genotype and the phenotype. And this is also, I would say, collapsing across sexes and age groups.
What about the fact that antisocial behavior was number 6 rather than 1?
I think antisocial behavior is much less likely to end up on your medical record than tobacco use disorder. It's a much rarer and more serious phenotype, and it's also there's different ascertainment biases for those two things in terms of being even in the medical record in the first place. These are people who have insurance, right? So there's selection against the Okay, so in terms of our bioannotation analyses, and again, these are in progress and we're open to suggestions, the basic, like, where in the brain tissue expression, it's in the brain, it's not in the brain. So we have significant signal in all brain-related tissues and no significant signal in no not brain-related tissues. I think this is important mostly because we're not picking up on, I did wonder if we were going to get testis, but we're not picking up on gonadal tissue. And we're also not picking up on things that might be grist for social biases here. So we're picking up on what we think we should be picking up on. In terms of the timing, we're finding significant enrichment in a very specific developmental window, which is the early to mid prenatal period, and not really in other periods of development. This is not the case for all psychiatric disorders. You don't always get this temporal specificity, and you certainly don't get that temporal specificity just in this early to mid prenatal period. I'm kind of skipping forward. In terms of cell types, we're seeing enrichment in GABAergic and dopaminergic neuron gene sets, signals observed in midbrain neuronal subtypes, and some fetal-specific gene sets. I think there is much more that could be done here. We also did one drug repurposing analysis, drug sets analysis. Just as a background, I just want to note again that for many of these diagnoses, there is actually no FDA-approved pharmacological treatments. There's nothing for conduct disorder. Conduct disorder, according to one study of global disability, ranked it as the number 30th most impairing disorder, full stop, not just psychiatric disorders, above autism, above malaria. It's the most common reason to be referred for psychological treatment if you're a child or adolescent. In both the Europe and the UK, and there's currently no pharmacological treatments for it. I think we can speculate about why that is. What is the investment in finding a treatment for a disorder that's characterized by moralized and disciplined behavior? So we did a drug sets analysis that identified 8 compounds as potential repurposing candidates. All of them were allosteric modulators of GABA-A. One of them I was particularly interested in is that's zuranolone, which is a synthetic form of allopregnanolone. FDA recently approved that for the treatment of postpartum depression. Women with ADHD are 5 times more likely to be diagnosed with postpartum depression than women without an ADHD diagnosis. And allopregnanolone is produced at the highest point in the human lifespan by the placenta in the early to mid prenatal period. There is some research on animal models of preterm birth, where male offspring who are born early are at elevated risk of aggressive and hyperactive behavior, and treatment with allopregnanolone at birth reduces that hyperactivity and aggressive phenotype. So we're finding this interesting convergence between our bioannotation, identifying with the prenatal period, our drug repurposing, and some of the animal models of this hyperactive, aggressive phenotype. Again, I think there's a lot to be done here. Okay, so some concluding thoughts. ADHD is defined in the DSM as a neurodevelopmental disorder, and everything else is not. And it just really has led us to think, what is a neurodevelopmental disorder? All day I've been talking to you, brilliant people, and many of you say, well, I study neuropsychiatric disorders, and then I say, what disorder? And it's usually autism, schizophrenia, Alzheimer's, Parkinson's, Rett syndrome. When I say neurodevelopmental disorder, no one thinks of the persistently aggressive child, the one who's constantly referred to their counselor before they get suspended from school. But when we look at genes that are associated with antisocial behavior, serious persistent antisocial behavior, we find genes that are also related to substance use, that are also related to ADHD, that are expressed preferentially in a very specific part of prenatal development, and that by any metric, I would consider a neurodevelopmental disorder. I do wonder, why is that? Why is it that there is so much investment in studying the genetics of autism or the genetics of schizophrenia, but not the genetics of children whose primary complaint is these social violations? And my hypothesis, and I can't prove this is conjecture, is that we have intuitions about the relationship between biology and blameworthiness. We're much more comfortable in a Pelagian framework where we can say ADHD is not a bad behavior, it's neurodevelopmental. But when we're thinking about children and adolescents and adults who persistently harm other people, it could be both. It could be both neurodevelopmental and harmful and violating our moral norms. And I think we have a lot of trouble holding those things in our head at the same time. And then finally, genetics is not staying within the halls of the university. There was just recently a very, I think, well done, for instance, New York Times Magazine report about polygenic screening for embryos. I was telling Rebecca about this earlier, I have been contacted by more than one defense attorney about genetics related to violent behavior. And we have already seen this happen. We've seen it happen with addiction, or I think we're seeing it happen with Ozempic right now, which is how does new technology get entered into the public debates about the moral ascriptions we make about a behavior. And I think it's a very open question how this emerging genetic knowledge is going to play out in the future. All right, I'm going to end there. Thank you very much to NIH. I hope that you still exist. The Jacobs Foundation, the Templeton Foundation, the fellow investigators of the Externalizing Consortium. I'm not on Twitter these days, but I am contactable by email. And my book, which is working title is Original Sin, The Genetics of Vice, The Problem of Blame, and the Future of Forgiveness, will be out next year. Thanks so much.
Short questions I have to leave, unfortunately.
Okay.
Number one, on the chart, there was a name for the first 3 quarters, but not the last quarter, where narcissism and all kinds of other stuff were there. What's going on here? Do we not have names for those things?
Like in terms of the.
I recognize many of my friends on the bottom.
So this is, gosh. There's few more controversial areas, not even ignoring psychiatric genetics, in psychiatry than the personality disorder. These were almost like gotten rid of in DSM-5. These are vastly under-researched, just generally, in particular in the genetic space. There was just one GWAS of borderline PD, and there is now a personality disorder psychiatric genetics consortium. I think that Bob Kruger put these here because it's like labels that people get when they tend to get in trouble with other people, but they're very under-researched. Yeah.
Can I ask a couple more really quick ones? If the law does not apply in the same beneficial way to everyone, for example, saying, oh, like you gave the example of staying later in school and stuff like that.
Yeah.
I'm just wondering, should we be, for example, should we be tailoring the treatment even in punishment, quote unquote?
Oh gosh, I love how you just phrased that as a quick question. I mean, I think that's been the dream for many people for a long time, which is you have a child or you have an adolescent that comes into clinic and they have this kind of constellation of problems, and you have first-line treatments, like let's do methylphenidate and parent training and like see if that works. And then only when you have that failure to respond to first-line treatments do people start to think, what else do we need here? There isn't a very deep bench of treatments and interventions, and particularly as people age, they're much less likely to end up in treatment and much more likely to end up in discipline.
But even in parenting styles, like how should I treat one kid versus another kid, for God's sake?
Yeah, I mean, I think that is a fabulous idea that we are very far from attaining.
Okay, perhaps one
Yeah.
last one.
Okay, I'm actually going to pause you and let.
Oh yeah, of course.
Yeah.
Becky, you had a question.
Rebecca.
Thanks, well, this was gorgeous. Thank you.
Thank you.
I have a kind of combination of a technical and a conceptual question that's
Yeah.
just a detail. So when you started here, you said the biggest distinction is externalizing versus internalizing.
Yeah.
And then at the very end, when you got to the drug study,
Yeah.
it was a drug for treating depression, which I thought was internalizing.
Yeah.
So I want to test this question at two levels. Can you explain how the drug replacement GWAS analysis works? Because that's not what I'm
Yeah.
familiar with.
Yeah.
And also, what do you make of the fact that what it came up with looks like the opposite
Yeah.
kind of disorder?
The drug sets analysis is basically given current compounds that we have, what gene products do they affect, and then how does that relate to the genes that have variants associated with our phenotype in the GWAS. So it's a relatively clumsy kind of triangulation. Postpartum depression is interesting because it doesn't fit neatly always with other internalizing. Women who have anxiety, for instance, are not necessarily more likely to get postpartum depression. Some of the best predictors are, like I said, ADHD. And then also, even though you have this distinction between internalizing and externalizing, they are still comorbid with each other. You also have general comorbidity. So, I think we're, the question of lumping and splitting, like, do I have internalizing, major depressive disorder, right? The correlation is around 0.5 here. I think it's a useful distinction, but I think if you're analyzing any psychiatric disorder, to some extent you're picking up on some genetic signal that's relevant to all of them. Yeah.
I want to ask a slightly philosophical question,
Yeah.
which is that, when we think about people with a propensity for behavioral disorders or criminality or whatever it is, they got that from someplace. It might be partly from genetics, and your studies show that, it's like 6% or something like that is coming from genetics, but it's coming from somewhere for every single person.
Yeah.
What explains the particular power of genetics
Yeah.
and explaining it, and how can we get past that for the
So
rest
this
of
is
the
a very,
cases?
Dawkins in the extended phenotype has an extended exposition of exactly this point, and he says, whatever you make of the thesis of determinism, like in the kind of philosophy of free will sense, the addition of the word genetic won't make a difference, right? And then he goes on to say what gave genes their sinister juggernaut-like reputation. So I think there is like one perspective, which is either you believe in determinism or indeterminism, and either you're a compatibilist or you're not. And like the genes isn't really relevant to any of that. what's interesting to me empirically is that you can make a philosophical argument for that, but people do reason quite differently about genetic causes versus environmental causes. Some people have argued that genes operate in a secular age as what people have called an essence placeholder. So it's no longer acceptable to talk about people having souls, but they still have a sense of that there is some sort of deep down singular true self, and now that's imputed to genetics rather than to, you know, a spirit that like God gave your body when you were conceived, or you know, whatever the kind of immaterial interpretation of it is. And I think that goes a long way in thinking about, like, you can imagine you if you had been severely abused as a child. That's a way of projecting yourself into someone's circumstances that can engender fellow sympathy for your fellow man. But who are you if you have an entirely different genotype? That is more difficult to imagine, and I think that kind of those kind of implicit counterfactuals when we're thinking about the relation between genetics, environment, and the self contribute to why we reason differently about genetics and environment. The other reason is I think we all now live in a country that was founded by Puritans, and guess what they really believed in? Original sin. They really believed that the thing that made you bad, and deserving of punishment was the thing that you physically inherited through your father's sperm. Like, that is how they, and I think that we can still swim in that intellectual environment. Like those are the people who set up our government and our carceral systems and our legal systems, and I think that's still in our thinking.
That was really great. So I really appreciated your emphasis on the point that, you know, like, best data sets, the most extensive knowledge that we have is really this very narrow population. And it was also great to see that you're moving beyond that, and then now we have data sets from multiple groups. And I wonder if you, so I guess, so one, maybe a very specific sort of close to the data question is, like, how similar are the, like what's the correlation of the beta weights across those groups, for example?
Yeah.
And then maybe another version of that question to try to push a little deeper is, like, the differences in those populations are both, like, there are sort of, there are aggregate, on average, physiological differences, other you know, the differences in the distribution of the genes in other ways, but there are also the differences in the social context. And then we can also start to imagine sort of similar similar populations, but, you know, Europe, different countries, and, like, what do we know so far about, like, the affordability of the generalizations of, like, the sense of beta weights that we're going to be able to predict as we move across contexts like that?
So setting genetics aside, what we know just phenotypically is that you see very similar rates of ADHD and cognitive disorder and very similar sex ratios across countries and across different populations within countries. People are much more likely to be socially punished or involved in the criminal justice system for those behaviors if there are some groups versus other groups. But if we're just talking about, like, do you have conduct disorder, you get a lot of similarity across population groups and across countries that have been studied, which are still not universal. If you're looking at just the factor structure, like, how do these things correlate with each other, compare with each other phenotypically, you also see really consistent factor structures in the different populations that have been studied in the United States, right? So if you're looking at Asian Americans versus African Americans versus white Americans, you see the factor structure is, like, pretty much the same. Things hang together in the same way. The cross-ancestry genetic correlation is an attempt to quantify into a single number, that sense of, like, or the genes that are associated with this factor in European-like individuals also associated, you know, in the same kind of scaling in the same way. And that was indistinguishable from one. That didn't really surprise me, given how similar the epidemiological evidence is across groups. I think, you know, we just need more data. I mean, there just needs to be so much more data. And I think any effort now to disentangle differences across genomic ancestry versus differences across environment, the kind of structure of environmental risk is probably premature. given how entangled and confounded those are and how much, how little data we have. Yeah.
I would like to come back to the idea of like maybe modifying punishment based on the idea of like some disorders can come from like genetic factors. But don't you think that it also can open a door to saying that some kids will not change? And like,
Yeah. So there's been a lot of work on this in social psychology about how does genetic information change people's perceptions of both blameworthiness, but also changeability or mutability. I think some of the best research has been done around body weight. So the more that people think that their body weight is genetically influenced, the less likely they are to moralize weight, the less likely they are to feel guilty or think that someone's done something morally transgressive if they're overweight. But they're also less likely to believe that weight can change over time. So some people have referred to this kind of like this double-edged sword of biogenetic explanations. To some extent, I think this is a meta-science question that needs more research. We as science communicators or as scientists are like, well, this is my best effort to describe the reality of both this being meaningful but not deterministic. But there's a reason why, communications as a field exists, and like the actual evidence about what framing of these results kind of emphasize one side of the story versus the other, I think is really understood right now. Yeah.
I have a question. I understand. It's not very well formed, but I'll frame it as a
It's my
comment
favorite
and
kind.
then a question for you. So I study a single gene disorder, Rett syndrome, which you correctly pointed out is a neurodevelopmental disorder. And I know several girls who have Rett, and I of course work with mice. Even though it is a single gene Mendelian disorder, every individual with a mutant gene, MECP2, will get Rett syndrome. I'm also impressed by the variability and how much even a mouse can be improved if you rear the mouse not in a little isolated cage, but in a rich environment.
Yeah.
So my question for you is, I know that you don't take the position of hard-nosed biological determinism when you have much history around discussing that. I wonder whether there will be a polygenic biological potential score as opposed to a risk score.
Yeah.
Because biology is as much about potential as about determinism.
Yes. And that's why some people have moved to the SSGAC's attempt to just abandon the use of the word risk score and call it a polygenic index. That was their attempt to sound very neutral. I do think that that old idea of kind of a reaction formation, that there's a score that represents a potentiality of outcomes or a range of outcomes. Part of that is a representation problem, a language problem. I don't have a good answer to that, but I agree with the principle. And I just want to end. I guess I want to supplement that idea with an empirical fact, which is there's a line of economics research on who is most likely by the age of 35 to have successfully started a startup in the United States. So that they are making money and they're self-employed and they employ other people. And the two predictors that were the best ones were having social advantage, so being white and coming from a high SES background, and two, being involved in delinquency and antisocial behavior as an adolescent. And I like that finding because I think that obviously risk-taking and norm violating can be harmful. I don't want to underestimate the harm of that. But also, I'm a Durkheimian here, which I think that like some element of deviance is necessary for society. Like none of us wants to live in a society in which only the most like puritanical and conscientious and controlled of us are allowed to, exist and do our work, right? So I think even within things we call a risk gene, because they confer risk for things that really can be very apparent. Like, if you look at that medical record, that is a record of pain and suicidality and infection and disease. And also, that variation can have upsides and can be necessary, can be socially valuable. But keeping both of those in mind at the same time can be really
Okay. One last question, Jill.
Thank you. I was curious about, you had such a strong bias to early brain genes expressed during early brain development, and I was wondering if you could say something about that, whether you think it's slower development or if there's any other thing you can say.
I don't think there's anything more I can say about it right now, and I will say that this is, the part of the project, the bioadaptation, is the part that I'm most intrigued by, and is the least in my wheelhouse and skill set, which is why talking to people here is so interesting. Yeah, I don't think that, other than, again, it doesn't look like that for every psychiatric disorder that you look at. And I mean, and one more thing, which I didn't have time, is if you do the FEWAS in the medical record by age epoch, so instead of pooling everyone together, you look at 0 to 10-year-olds, 11 to 20-year-olds, it's like 19 to 25-year-olds. What you see in childhood and adolescence is conduct disorder, ADHD, mood disorder, bipolar. It's basically like psychiatrists being like, something's going on with this kid, and I'm putting various labels on it. If you look at adulthood, it's antisocial behavior disorder, substance use disorder, tobacco use, and the medical consequences of substance use, right? In that 19 to 25, the top 10 out of 20 EHR codes that are associated with externalizing are complications of pregnancy. Early labor, threatened labor, fetal abnormality requiring management of the mother, and that's even pooling both sexes. So there seems to be something both in the genes predicting mom's prenatal problems and genes being expressed in the prenatal period that is really, there's an interesting story there that we just haven't teased out yet.
Okay. Thank you. That was
Thank you so much.
fantastic.